So as we've discussed in the previous webinars, for those of you that are new to the series, the Apical laboratory was founded in 2016, rapid processing in order to achieve fast results for the growers. We've unpacked a number of different unique methods, both from sampling protocol to laboratory protocol to reporting data structure and so on and so forth, in order to redefine what it means to learn about your crops, interact with them on a day-to-day basis, and make crop improvement from that data. Doing so, the suite of tools, leaf, soil, and water mineral analysis. We time and time come back to this concept: nutrient balance builds crop resilience, and you'll understand really what that truly means by the end of this webinar, hopefully, anyways. As we continue to discuss the toxicities within the crops that are either natural or induced, listed here: aluminum, sometimes potassium, sometimes sulfur, sometimes nitrate, oftentimes chloride, significantly oftentimes sodium. And the problems that they bring when they're inside a plant at overabundance amounts or in overabundance. So, attraction of pest and disease, nutrient lockout, poor fruit quality, petal or fruit abortion, root issues, poor sugar, protein development, et cetera, et cetera. So if any of these are of interest to you in your cropping system, I encourage you to really understand what the 10 principles are all about, because that's how to get back to from where you are today to having crop resilience through nutrient balance. So without any further ado, we will get into the 10 principles here as we have really unpacked. And so the number one and most important thing to understand is technically covered by the definition of an autotroph, which is what a plant is classified as. But essentially, plants can thrive in nature on low levels of nutrients delivered from soils by microbes. That is a very key principle. If you go to any national forest, you'll realize this. If you go to a uncultivated fruit tree that has been overlooked for a bunch of years, you'll probably see a good amount of fruit still on that tree. If you go to, drive down any sort of highway, look at your yard. The list of examples just goes on and on, where plants are thriving in nature on low levels of nutrients from soils delivered by microbes that have no fertilization for maybe in their entire lifetime. Most plants, in fact, on planet Earth don't receive fertilization, and they thrive on low levels of nutrients from soils delivered by microbes. So it's a really key point. And, oftentimes in our rush to, want to grow the best crop, we oftentimes forget that nature has been growing crops for thousands of years with or kind of without us, or, in some situations, in, a variety of, partnerships with humans. But at the end of the day, these plants can grow in nature without us. Now, how successfully they grow is really dictated by the environment that they're grown in. And this is I know other agronomists out there talk about this, instead of trying to shift the plant, right, you shift the growing environment, right? Well, I think there's something to be said for both. And oftentimes when we see a leaf analysis come in, and then we try to affect both the plant and the soil through our detailed recommendations, we see those as having the most impactful and most rapid and most profound plant response. So number 2, plants in nature generally maintain elements that are very balanced between new growth and old growth. This is key. This is extremely key point, because if any of you have ever gotten a SAP analysis from us, chances are there's going to be imbalances all over the map, right? All over the report. And you're left wondering, what do those mean? What do those highlights mean? How is that affecting my crop? How is it limiting my crop? Et cetera, et cetera. And you may end up with more questions than answers. For years, we really dug into our detailed comparison report and studied how nutrients flowed and were proportional to other nutrients, both in the new leaf or in the old leaf, and so on. And through that time of study, we gain significant insight into nutrient antagonism, patterns of accumulation, sources versus sinks, where nutrients would start from to flow and end up, and so on and so forth. Nutrient interaction from one nutrient to another, and so on. And when you're looking at data sets like this that we have, like we have here as a team, you know, you might look at, if you're a grower of ours or even a consultant, a couple of tests or maybe even a couple of hundred tests, per season. But, we're looking at a couple of hundred tests, oftentimes per week or sometimes per day even. So the data set that we're looking at here is significantly expanded from what a lot of the other growers are looking at, or what a lot of the other consultants are looking at. Additionally, the data structure that we have employed has elements of it that no other lab has ever done, right? No other lab that I know of has gone to the trouble to, group elements that might compete with one another on the same report, or compare and contrast, new leaf to old leaf in terms of percentages and gradients and so on and so on to track nutrient flows. So once we realized that nutrients were starting to flow and that this balance was what we wanted, what we're kind of after, that whole system was unpacked in order to help growers find that balance. And in finding that balance, what we found is we're able to unlock tremendous value for the grower, for the agronomist, for environment, for the crop, and for ultimately the consumer. So, getting back to element ratios and nutrient antagonism within plants, you have always known, dating back to, the 1950s and the work of William Albrecht, and probably even before him, there's significant influence on element to element ratios within soils. What we found is that this situation actually is compounded when in the plant. So in other words, where you might see a small ratio out of balance in the soil, we see that hyperaccumulate in the plant. And when we look at growers' soil tests, and then we look at a plant test, and we look for correlations, oftentimes, especially in perennial crops, we see that a nutrient may start out as an excess in the soil, but then hyperaccumulate into a crop killing excess in the plant. That crop killing excess in the plant may survive year over year in perennial systems, dragging down the entire productivity of a farm, a cultivar, an entire bioregion, entire country, right? And without knowing it, right? And so if you start to unlock these nutrient values and the ratios and the hyperaccumulations of nutrients in different parts of the plant, you will now be able to gain insight into where that overaccumulation is influencing other elements. Because what we find is when the plant hyperaccumulates one element, it hyper underaccumulates another. And that hyper underaccumulation causes a cascade of imbalances, malformed proteins, unstable DNA structures, poorly formed sugars, and overarchingly lack of vigor or full capabilities of crop production. Carbon is king. Thanks again very much, and look forward to seeing you all next week. Take care.